Hello, biologists. Welcome to lecture. Today we're going to talk about gas exchange and circulation. And so I put an image of this creature for the opening slide. Do you recognize it? It is an oxalotl. So an oxalotl is a salamander. And what's notable about it is it has these external gills instead of internal gills like a fish. And so we can see the respiratory organs really clearly, and we can see the blood vessels that are running along them. So here, this fringe, it's thin to increase surface area, so structure and function going together. And this is where we have diffusion of oxygen from the water into the bloodstream and diffusion of carbon dioxide from the bloodstream into the water. So this is our site of gas exchange. I also think these critters are adorable. They're from Mexico, around the area of Mexico City, and they still live out in the wild, even though their habitat is endangered. But just the cutest little critters. So let's get into it. How do other organisms do this? If gills are not usually internal, what are they? What are lungs? Let's talk. First, if you're small, like this planarian, one of our platyhelminthes, one of the flatworms, or unicellular, you can just use diffusion. Like if your surface area to volume ratio is low enough, diffusion works fast enough that you can get rid of your waste and get in oxygen, all at a rate that keeps you alive. So planarian like this, it just diffuses right through their bodies. They don't have a circulatory system. They don't have blood. They don't have a heart because they don't need to pump fluids around their body to transport gases. Instead, it does it all on its own. We, however, are mostly multicellular animals, and so we have to have other systems to make it work for us. One of the systems that we use is the tracheal system. The tracheal system, trachea for like tube, is what a lot of insects use, not all of them, but a lot of them. And it's this network of holes on the side of their body that attach with tubes to longer tubes that run through their entire system. So here are the little holes. Those are called spiracles. These are the breathing holes. They suck air in through here. Those spiracles are interconnected to these sacs, and these sacs allow the air to diffuse through their body. As they move, as they wiggle, that helps push the air through and around. Bumblebees will intentionally scrunch up their abdomen to help move the air around. Not all bugs do that kind of ventilation system. There are little muscles involved, but these tubes are what move the air through. So this is your tracheal system. Unlike the gill system, so here we have a pike. This is a type of fish. We've pulled back the gill cover, also called the operculum. And under the operculum, you can see these gills in your fish in the pharyngeal region. These gills, much like the oxalotl, they have a lot of fringe, and that fringe increases the surface area. And so we have capillaries, which are blood vessels running through this area, and gas exchange occurs here. So blood comes in. That's why they're so pink and red, is they're full of blood. That blood moves across the surface area of the gills. Oxygen diffuses in, carbon dioxide diffuses out. This is a passive process. It's just the moving down your concentration gradient. And through that, then the fish uses its heart to pump that blood around its body. So no lungs, gills instead. Here's a close-up of gills. We have a beautiful fish here. This would be the gill cover or the operculum, where you'd pull it back and look inside. You would see the filaments of the gills. In those filaments, we have blood vessels running through them, bringing a lot of carbon dioxide to diffuse out and oxygen to diffuse in. Then they'll go back to the body. These little filaments are stacked up on top of each other like a lot of little fringe. And if we were to look even closer, we would see that there's plates on these filaments. That's going to increase the surface area even more. Here we have oxygen-rich blood. Where's the oxygen coming from? It's coming from the water that flows through these plates. So lamella means plate. So as the oxygen moves through, oxygen is diffused in. And then here we have a lot of carbon dioxide in the oxygen-poor blood. Carbon dioxide will be pulled out as the water moves through. We use lungs. So instead of using gills or a tracheal system, we have one major breathing tube. Our major breathing tube is called our trachea. Our trachea then branches into bronchi, plural. And then we have more branching into tinier tubes that are called bronchioles. And then those will attach to sacs where the air exchange happens. It's on the next slide. All of this white that's around it is going to be cartilaginous rings that keeps our tube open so it doesn't collapse. We want to be able to breathe at all times. So those are rings of cartilage around your trachea, keeping it warm. Keeping it open. We also are going to keep all of our air inside as we do gas exchange. So to remove all the particulate is important, to humidify the air is important, to warm the air is important. Otherwise, we damage these sensitive tissues. Inside of your tracheal system, inside your upper respiratory tract, you're going to have cells that assist with that. Everything is coated in a nice layer of mucus to keep things wet. There are cells with flagella and cilia that are pushing up towards your throat, and that will catch particulate in the mucus and then send it up to the area where you can swallow it. So we want to get that junk out of our lungs, and our little cilia and flagella are helping us push it out. Here is our trachea extending to our bronchi, and then our bronchi branch into bronchioles. At the ends of these bronchioles, where it's not really being shown here, are alveoli. So this is a bronchiole, this is a bronchiole. And then attached to the end of all of these bronchioles, there would be sacs of membrane. Now this membrane is made of simple squamous epithelium. It's very thin. It's thin for gas exchange. When you breathe in, these little sacs inflate, and they never really deflate, but they inflate even more. And so that oxygen will be able to just pass right through that single membrane of the simple squamous epithelium. It's coated in capillaries, our tiny blood vessels. Those capillaries are picking up oxygen and releasing carbon dioxide. So here's oxygen getting sucked into the bloodstream, and here's carbon dioxide leaving the bloodstream. You will then exhale the carbon dioxide, inhale another breath with even more oxygen. All of this is because of the concentration gradient of these molecules. So if you were in a space where the concentration gradient of oxygen in the air and your blood were the same, you could breathe all you want, but you wouldn't have diffusion occur. And so you wouldn't be able to get oxygen into your system. Now, what is respiration? Respiration is the process of breathing. In human lungs, your lungs are in negative pressure, and that allows you to draw air in. When you want to exhale, you're going to have to push that air out. So sucking air in is kind of passive, exhaling it is more active. That is why we care about making sure our lungs don't get punctured. If your lung gets punctured, you ruin that negative pressure and you can't suck in air anymore. So a punctured lung is a concern because it ruins the pressure difference. When you are moving air in and out at rest, we call that the tidal volume. I like to think about the tide moving in and out of the ocean. The amount of air that you can move in at max when you're forcefully exhaling after you've inhaled, that's called the vital capacity. So two different levels of air that we move through. Lungs of birds are incredible. They are the most efficient lungs in the animal kingdom. It's an incredible process. I have a YouTube video. I think it's so cool. It takes 4 breaths for the lungs of a bird to go through a full cycle. Our circulatory systems are how we move blood to effectively transport oxygen. So your blood has a lot of different functions, but one of its main functions is to carry that oxygen over to your tissues that are doing cellular respiration. Diffusion is not enough. We have to transport that blood everywhere so that each of those tissues can get it, because we're just too big for diffusion to work. Now, some of our arthropods and mollusks are going to have an open circulatory system. So this is called an open circulatory system because the blood vessels don't fully close. Instead, when the heart pumps blood, it pumps it to a network, and then it oozes out of the network, and the blood is referred to as hemolymph. Now, lymph is like a fluid that coats your cells, and it's not called blood because it's not contained. Hemolymph, heme for blood, is kind of a mixture. The interstitial fluid between the cells in a bug is no different than any other interstitial fluid. So there isn't blood per se. The tissues that need oxygen just get this fluid rained through. Then it's all collected back up, sent to the heart, and pumped out again. So this is why it's called open, because there isn't a connection between the blood vessels. The fluid that's being pumped is not contained. It diffuses through all the tissues and then gets sucked back up. So it works for them. It's an effective system for them. Everybody else, all of our vertebrates, they're going to have a closed circulatory system. It's closed because we do not have the blood leaving the blood vessels. The blood vessels are entire, so blood is always in a tube. Instead, the oxygen that we need has to diffuse out of the blood vessels. So here we have a fish. A fish has chambered heart. It's going to pump. It's going to send blood to the gills where it's going to pick up oxygen. That oxygen-rich blood will then travel to the body through a set of arteries and capillaries. So this will move the oxygen-rich tissue or oxygen-rich blood to all the tissues that need it. Diffusion is occurring everywhere along the way. It looks like it happens once, but it's really diffusing all over. And then when it has less oxygen in it, because you've given that tissue, you've given that oxygen to the tissues, and you have more carbon dioxide because you've picked up carbon dioxide as you go, then the blood goes back to the heart and is pumped to the gills to pick up more oxygen and release the carbon dioxide. Reptiles are going to do a similar process, but their hearts are more complex. So they're going to have a chambered heart as well, but now it's going to be divided into atria and ventricles. So let's start at the heart. The heart is going to pump blood up to the lungs. This is where gas exchange occurs. Pick up oxygen, get rid of carbon dioxide. Then it goes back to the heart, and the heart pumps it again out to the rest of the body. Now it's going to do diffusion at the capillaries, and it's going to come back to the heart when it's oxygen poor, full of carbon dioxide, and it'll get sent back to the lungs. There's also a connector where intermediate amount of blood that has like not the most amount of oxygen, but not the less amount of oxygen, is just sent back into the systemic circuit, systemic being the body circuit, not the pulmonary circuit. Note that this color is kind of a mix between blue and red, so it's a little bit of a purpley color. That's because these two chambers are not effectively divided. So this connector here, when the heart pumps, it pumps blood from both the oxygen poor side and the oxygen rich side out to the systemic circuit. Now, mammals and birds, we have a four-chambered heart that is completely enclosed from each chamber from each other. When you have oxygen poor blood, it comes in from the body. It's full of carbon dioxide. You're going to pump it to your lungs, get rid of the carbon dioxide, pick up oxygen. It comes back to your heart. Your heart will pump again, and it will push all of the blood out to your systemic circuit, your body, where you'll have diffusion occur, and then it will flow back to your heart to pick up more oxygen and then go back to your body. So I talked about this with a kid once, and the kid asked, so all your blood moves in like one chunk, like it goes to your heart and then pumped to your lungs? And I was like, oh no, it's like a continuous circular river. So it's never like one chunk of blood being pushed. You are full of blood at all times through all of your blood vessels. But it was an interesting misconception that I hadn't thought about before. So your heart is never empty. It's always full of blood, and it's causing pressure waves in your blood that is full continuous. So you're never empty waiting for more blood to come. You're waiting for like a tidal wave of blood to come from your heart. We talked about an open circulatory system when we were looking at a diagram. Now let's see what that looks like on an actual arthropod. Here is our representative of an arthropod, our beautiful honeybee. So this honeybee is going to be taking in oxygen through these openings on the side of its body called spiracles. It's not showing you the whole, but they line the thorax and the abdomen, and they suck in oxygen through these spiracles. Those spiracles connect to those tubes that you already saw in the grasshopper, to those air sacs, and they get pushed around the entire body. Now, we also have a circulatory system. As the air is moving through the tracheal system, you're going to have a tube of blood vessels, and this is going to be surrounded by many hearts. Each of these is a heart. These hearts are not really chambered. They're like pouches with circular muscles that squeeze and are going to push the blood around. The blood, it's called hemolymph. It's not true blood, so it's hemolymph. And in your heart, there's going to be a hole called an ostia. That is where the hemolymph drains back into the circulatory system. So I have fluid here. This fluid is draining into this hole, into this circulatory system. This is going to pump, and as it pumps, it's going to move the hemolymph out into the body tissues. It's going to become part of the interstitial fluid. So it's just going to bathe all of the cells in this bug, and then it will get reabsorbed back in through these holes, these ostia, and then it'll get pumped out again. So much like a closed circulatory system, but now we don't have capillaries connecting to paths. Instead, it just pushes the fluids out, sucks it back in, pushes the fluids out, sucks it back in. The fluids percolate through all of the tissues. And because it's not enclosed, it's called open. And because it's not blood per se, it's called hemolymph. Let's check out how a gill circulation works. So fish have a true chambered heart, and it has two chambers. It has an atrium and it has a ventricle. Atriums are on the top, and I remember that because I think A for attic. So attics are on the top, atria on the top. Atria is plural, by the way. Atrium is singular. So here's the atrium. In this fish, you're going to have oxygen poor blood coming from the body. You're going to pump it through your heart. Your ventricle is the stronger part of your heart. It's going to do the biggest pump, and it's going to squeeze it out to your gills. In your gills, you have those radiating gill filaments filled with gill lamellae, those gill plates. That's the site of gas exchange. So you're going to have tons of capillaries in your gills. That gas exchange is going to pick up oxygen, get rid of carbon dioxide, and it's going to flow through your body through an artery to a capillary bed. Arteries leave the heart. Veins come back to the heart. So artery, think A for away, attic and away. And capillaries are going to be the thinnest blood vessels. That's the site of gas exchange. So we have gas exchange occurring in the gills, and we have gas exchange occurring in the system, in the body. So all the muscles of the fish. The body we call the systemic circuit. So systemic circulation is taking blood to the body tissues. We're going to end up with more carbon dioxide and less oxygen. Then we're going to take that back to a heart with a vein. Think veins flow in. The word in is in vein. And your heart will pump again. The entire process will pump again. This is going to be an amphibian circulatory system. They have a 3-chambered heart, and they have double circulation. By double circulation, meaning they have a pulmonary circuit and they have a systemic circuit. So let's check out the heart. The atria are on the top. So there's 2 of them, but there's only 1 ventricle. So those are your 3 chambers. Oxygen poor blood is going to come into the heart, atria first, then the ventricle. The ventricle is going to squeeze and it's going to pump blood into 2 paths. One path is going up to the lungs, and it's going to do gas exchange at the capillary. Now, this is also going to be in the skin of the amphibians because amphibians can breathe through their skin. So they're going to have capillaries along their surface as well for gas exchange. Those blood vessels that picked up the oxygen are going to go back to the heart. So this would be your vein in, even though it's red, it's just based on direction. Through the atria back to the ventricle, and the heart will pump, And now the oxygen rich will leave the heart. So this is going to be an artery, come to a capillary bed for gas exchange, and then go back to the heart. So this is a vein into your 3-chambered amphibian heart. This is our reptile circulatory system. In reptiles, we have a 3-chambered heart, but they have 4 rooms in their heart. There are 2 atria and there are 2 ventricles. So it feels like it's 4-chambered, but it's not, because the 2 ventricle don't have a separation. So there's 4 spaces, but only 3 chambers. Let's follow the path of the blood. You're going to have oxygen poor blood coming from the body to the heart. First the atria, then the ventricle. You're going to pump that out to your lungs. We have a right and a left path. You're going to pick up oxygen, get rid of the carbon dioxide, and then you're going to come back to the heart from both lungs, so a right and a left path. Then your heart is going to pump out the blood to your body. So we have a right and a left path going to our systemic circuit. We're going to do gas exchange, and we're going to bring the blood back in. Note that it changes color. So blue for oxygen poor, red, kind of peach to me, for oxygen rich. But then the 2 mix together because your ventricles are not separate. And so what goes out to the body is a mixture of both. And so it's kind of a purple cross between red and blue. Now we have mammals. Mammals have a full 4-chambered heart with separate atria and separate ventricles. We're going to have a 2-system circuit. So we're going to have a pulmonary circuit and we're going to have a systemic circuit. Let's walk through the path of it. We're going to have oxygen poor blood coming in. So this is a vein, first to the atrium, then to the ventricle. It's going to get pumped to the right and left lungs. So 2 paths. You're going to pick up oxygen, release your carbon dioxide, and then blood is going to come back in from 2 lungs. So there's 2 paths back to the ventricle. And you're then going to go to the atrium and then to the ventricle. We're going to pump again, and the blood is going to leave the heart in an artery for away, and go down to the systemic circuit for gas exchange. We're going to talk about the anatomy in the heart in a second. It's pretty neat, and we'll get to label all of the parts of it. To see a review of what we just discussed, we have an amphibian heart, which has 3 chambers. We have our 2 atria, and we have our ventricle. It's going to pump blood up to the lungs, back to the heart, and then out to the systemic circuit, back to the heart. We have turtles, which are an example of a reptile. They have a 3-chambered heart, but now it's more separated. The ventricle is starting to separate off. They're going to take blood in from the body, pump it through to the lungs, back to the heart, and then out to the systemic circuit. But remember that because those ventricles are not fully divided, the blood that goes out to the body has oxygen rich and oxygen poor. The most oxygen rich blood comes right back into the heart, into the ventricle, and then mixes up before it goes out to the body. We have a full 4-chambered heart. By we, I mean mammals. Birds also have this. In this full 4-chambered heart, our ventricles are completely divided. Our atria are divided. When the ventricles pump, these little valves will close. Oxygen poor blood comes from the body to the atria, then the ventricle, where it goes to the pulmonary circuit. From the pulmonary circuit back to the atria, to the ventricle, to the systemic circuit. What is blood? And we're talking about blood in mammals now. So blood itself is classified as a connective tissue. It's a connective tissue because it cells in a matrix. The matrix here is plasma, and the cells are red blood cells, white blood cells, and platelets. If we were to spin our blood and separate out the solids and the liquids, we would find that almost all of it's 92% water. But if we spin it, about 45% of it would be our hematocrit, which is going to be our red blood cells. That's still a lot of water in there, but our red blood cells would make up 45% of it. The buffy coat is where we're going to find our white blood cells. And then the plasma, that's 55% of our blood. That's going to have all sorts of nutrients and hormones and ions and proteins. If you were to pull those goodies out, it would be called serum, but with the goodies in there, we'd call it plasma. Red blood cells are designed to be carrying oxygen. That is their goal. Their real name is erythrocytes. So erythrocytes. You have about 5 million per 1 microliter of blood. So it's a huge amount. Hematocrit, again, is the amount of red blood cells in whole blood once you spin it down. And if we look inside of a red blood cell, we're going to see that there is no nucleus. So we describe this as a biconcave disc. It looks kind of like a bialy or like a donut that you haven't pushed all the way through. And inside of this biconcave disc, you're going to have just millions and millions and millions of these proteins called hemoglobin. So a hemoglobin has quaternary structure, meaning that each one of these is a separate protein, and together they have a full 3D structure. So primary structure is the sequence of amino acids. Secondary structure is curls or pleats. Tertiary structure is the overall structure of this one orange one. And then hemoglobin contains more than one part of a protein. And so the quaternary structure is the full structure of these four working together. In hemoglobin, you have an ion of iron in a molecule called heme. This has a high affinity for oxygen. So this heme right here, that's where oxygen binds. And the iron is an important part of it, having the ability to bind oxygen. Your red blood cells only last about 120 days, and then you have to replace them. You make new red blood cells in your bone marrow, and you dispose of them in your pancreas. So you're going to be constantly cycling them out and making new ones. Our white blood cells, leukocytes, are incredible. They're used for our immune system. They're less than 1% of your blood, and they're way bigger than red blood cells. Now, what's really cool is they're not confined to your capillaries. They can actually leave and migrate into the tissue. We have 5 types. We have neutrophils, which are hunting for foreign particles to destroy. We have monocytes, which we're going to use to clean up damage. We have eosinophils, which are going to be used to kill parasites and cancer cells and are also involved in allergic responses. We have lymphocytes, which are going to be used to make antibodies. And we have basophils, which are involved in allergic response. Here is our mammalian heart. It is so cool. So we are going to have 2 sets of veins bringing blood into the body. So when you see the vena cava and the other vena cava, those are both veins. And we know that they're bringing blood in because they're going toward the heart. So that is where oxygen poor blood is going to be entering our heart. Oxygen poor blood is going to fill the atrium, and your atrium is then going to contract, and it's going to push the blood into the ventricle. It's like a little top off, because as your atria is filling with blood, it kind of pours down into the ventricle anyway. So your atria is going to contract, and then your ventricles are going to contract. Here, blood pulls through, and it fills this ventricle. Look at this pink right here. This pink is the muscle tissue of your heart. You're going to have a layer on the outside made of connective tissue called the pericardium. That's your heart sac. It's actually really tough and fibrous. But the pink itself is the muscle tissue. And we have a thin little bit of muscle here. And then we have way thicker muscle down here. And that's a good structure and function example. Your atria, when they contract, they're just topping off the ventricles. They don't have to send blood very far. Your ventricles, however, when they contract, they're going to have to send blood a lot further, and they're creating a lot more pressure. So your right ventricle, and it's named right because it belongs to the person you're looking at. So it's their right side, even though when I'm looking at this picture, it's my left that I'm talking about. If I were to turn around and talk about my heart, it would be my right side. So it feels backwards because it's whoever owns the organ, we use their right and left. So on the right ventricle, we're going to squeeze, we're going to contract As we contract, that's going to push close this valve so blood cannot go backwards. And we're going to push open this valve and we're going to send blood out through this artery, A for away, to the pulmonary circuit. So this purple branch on both sides connects to a lung over here on the right and the left. Let's talk about some anatomy in this right ventricle. We have these muscles here. These muscles are called papilla, and they have these little cords attached to them that attach to this valve. That's so that if your heart pushes, these little valves don't flap back and forth like a saloon door. So we don't want them to go past their meeting point. And so you have these little tiny tendons, they're called chordae tendineae, and they're going to hold it in place. We also have a bunch of little ridges and bumps inside that's just muscle fibers together. And so that's going to be the anatomy of our ventricle. We have our AB valve right here. So atrioventricular. And this one is on the right. This has 3 flaps, and so it's called the tricuspid valve. So 2 different names for this valve. We're also going to have semilunar valves. So SL semilunar. They look like little half moons. They're also called the pulmonary valve. So we're going to have right and left semilunar valves. Those are going to close when the blood starts to backflow. So it makes like these little catchers, and it's going to parachute out and shut that valve once your ventricle is done creating a force. So it's going to prevent backflow from the pulmonary artery into the ventricle. So I'm pushing blood into my lungs. I'm doing gas exchange. Now I want to get that blood back because I want to send it to my body. It's going to come back through the pulmonary vein, so vein for in. These are shown in yellow. Often this would be shown in bright red because this is full of oxygen, but that's okay. The color doesn't actually change in your blood. Your blood is always red. It's just like a brighter red or a more darker red. So like raspberry versus burgundy, but it's never blue. So I wish we had chosen different colors to show it when we started doing this trend, but we didn't. So these ones are yellow. That's okay. This is showing you oxygen-rich blood coming from the lungs to the heart, so it's a vein. They're going to pool in the left atrium. The left atrium is going to kind of overflow, and then it will contract with its thin little muscle, topping off the ventricle. The ventricle is now full of oxygen-rich blood, and I want to do the hardest pump possible because I need to send that blood all the way out to my head, to my arms, to my legs, to my toes. So I need to overcome a lot of resistance of blood pressure within my body. And so my left ventricle here is super strong. Like this is a good amount of muscle, but this is way thicker. And we're going to see diagrams of this too, where it's really showing you how thick the left ventricle is because it needs to make a larger force, a stronger contraction. The left ventricle also has these muscle fibers attached to the tendons to hold your atrioventricular valve shut so that it doesn't flap like a saloon door. This one only has 2, so it's often called the bicuspid or the mitral valve. Mitral refers to a hat, and so someone thought it looked like a mitre, and so it's often called the mitral valve. Bicuspid is good. Left AV valve is good too. Make sure you remember that AV is atrioventricular. So as this pumps, you're going to increase the pressure here. It's going to force this valve closed. Your little chordae tendineae are going to hold it in place so it doesn't flap back and forth. And that means that your blood only has one place to go. It's going to go out your left semilunar valves, out through the aorta. So we call this the aortic valve. And it's going to go from your aorta up to your arm, up to your arm, up to your head. And then this tube continues down behind you and takes the blood to your torso and to your legs. So cool. Let's look at another image. This one is showing us the path of the blood. We have the blue and red scheme happening. So we have the superior vena cava and the inferior vena cava. Both are veins bringing oxygen for blood in from the systemic circuit. We're going to fill the atria. The atria is going to contract, topping off the ventricle. This happens on both sides at the same time. So as my systemic system is sending blood back in, The pulmonary system is also sending blood back in. So here's my pulmonary veins, both sides. Blood comes back from the lungs into the left atrium, and it will be contracted to top off the left ventricle. So we're pooling in the ventricles. Our next step is the contraction. That's going to push blood through. Our ventricles are now full, and we are going to do ventricular contraction, and that's going to shut our AV valves, our atrioventricular valves. The muscle is squeezing. The blood pressure is rising. It will overcome the pressure needed to go through the semilunar valves. These are taking the blood to the lungs, and this one is the aortic, taking blood to the pulmonary circuit. So now under we have some vocab. In cardiac dystole, or diastole, no dys, diastole, all chambers are relaxed. When you have systole, that is when things are contracting. So here, our atria contract first, and our ventricles are still relaxed. That's topping off the ventricles. Next, our atrias are going to relax, and our ventricles are going to contract. But the blood does not flow backwards because we have these valves, the AV valves, that are held in place with those little tendons. Once the blood is pushed through, there might be a release in pressure and it could fall backwards, and your semilunar valves will close, keeping the blood from reversing. Here we have the same process shown all at once. Each part is labeled, and now we get to see into the heart tissue itself. So we have this muscle here, and it has a couple layers. The inner layer of the heart muscle is called the endocardium. The thick part of the muscle that does all the squeezing is called the myocardium. Then on the top, we have a layer called the epicardium. So this is your heart wall. Then we're going to have a space. This is a pericardial space. In this pericardial space, you're going to have lubrication, because your heart is moving against its sac, and movement creates friction, and that would cause a lot of heat, and it would create damage. So you have a space around your heart that's filled with like this really, really greasy fluid to keep it nice and lubricated. Then you have your pericardium. Your pericardium comes in two pieces. You have the fibrous pericardium and the parietal pericardium. If we think about the pericardium as everything, it's actually also including your epicardium. So all of this is connective tissue. And then below that, you have your layer of muscle. All right, this would be a good slide to pause the video and see if you could name all the parts of the heart on your own. So a little bit of recall. Let's talk about how this is controlled. The control of the heart is called the cardiac cycle. Your heart contains autorhythmic fibers. When we say fiber, we mean neuron, that are self-excitable. That means that your heart keeps its own pace, regardless of what the rest of your body is doing. That doesn't mean that it's not talking to your body. When you're in your sympathetic division of your autonomic nervous system, your body's going to tell your heart, we're scared, we're worried, we need to fight or flight, and then your heart will respond. But your brain doesn't have to tell your heart to beat every second. That's what they mean by self-excitable. So the two are connected for sure, but your heart is doing its own setting of its pace independent, even though it still talks to your brain. So let's talk about how we send our electrical impulses through our hearts. We are going to start with our sinoatrial node. This is the one that sets the pace, so it's called the pacemaker. These cells are often referred to as pacemaker cells. And when these cells depolarize, they send an electrical signal that starts at the atria and causes a depolarization event through these neurons. That will set off the next set of neurons involved in the process. That's called the atrioventricular node. That atrioventricular node will further propagate the signal. The signal will move down here through these sets of fibers called Purkinje fibers that move to either side of the ventricles, and those are going to have your ventricles contract. So up at the SA node, that's where we set the pace, and the process starts there. The electrical impulse goes from the top to the bottom. So our atrium depolarize first, contract first, then our ventricles depolarize and contract. And so that makes sure that our atria contract, then our ventricles, atria, ventricles, atria, ventricles. If these things try to pump at the same time, the blood wouldn't effectively move. So the timing is important and the rate is important. When you're listening to a heart in a stethoscope, or you have your heart on, your head on someone's chest, or like I listen to my kitty's heart all the time, you can hear two sounds. We call it the lub dub. So it goes dun 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 dun. The lub specifically, the first sound is a little bit smaller and the dub is a little bit bigger. That's because the lub is your AV valves closing and your dub is the semilunar valves closing. And so you can hear both of these processes. With a stethoscope, you can even sometimes hear the blood swooshing back and forth. We can check whether or not this system is going as it should using a electrocardiogram, also called an EKG. The reason it's a K instead of a C is because the term was originally German and they spell cardio with a K. It's also called an ECG, but it's more commonly called an EKG. Here's how it works. You're going to have your heart and we're going to be measuring the electrical impulses as it goes through a cardiac cycle. This first little bump is when the sinoatrial node gets its information and it causes the atria to contract. Then we have a little break. That's when your AV node is being activated. Then the AV node sends the information down to your ventricles. Your ventricles get ready to contract. We're going to have a big electrical activity event, and then everything resets. So that spike that you see on the EKG machine, that's for ventricular contraction, and the little bump before it, that is for the atrial contraction. Now that we've talked about our whole systemic system and our pulmonary system and our hearts, the different types of hearts for animals, we also need to talk about the characteristics of blood vessels. So blood vessels come in three types. We have arteries that take blood away from the heart. They're going to have more pressure in them than the other types of blood vessels. The reason they have more pressure in them is because they are taking pressure from the contraction of the left ventricle, which has to squeeze blood all through your system. So that's like the hardest pump possible. Because of this, arteries are going to be thicker walled. They're going to have more elastic tissue. They're going to be preventing any ruptures. Those arteries are going to branch into smaller tubes called arterioles. Arterioles are going to go through all of your body, so everywhere, and they are going to branch even further into capillaries. So capillaries are the thinnest blood vessel. They are simple squamous epithelium. They are one cell thick, and they are so thin that gas exchange just occurs right through the capillary membrane. Capillaries are actually so small that red blood cells can have trouble traveling through them. And so red blood cells stack up into these little packets as they move through. They're called rouleaux, and it's pretty cool. So capillaries, this is where gas exchange is occurring. Then your blood is going to go back to your heart through a small tube called a venule that connects to a vein. Now, veins are a lot less pressure filled. These are the ones that they take blood from when you're donating blood or getting a blood test done. They do not stick a needle into an artery because that's way higher pressure. You take it from a vein. And the veins, because they have lower pressure, they're thinner walled. But we also have to worry about them backflowing because there's thinner pressure. So in your legs where you have these really big, thick veins, and they need all of this blood to get back up to the heart, they're going to have one-way valves in them. So as you move your body, you create what we call a muscle pump. And that muscle pump pushes blood into your veins and squeezes it upward, kind of like squeezing a tube of toothpaste. These one-way valves are going to prevent backflow. So as you move your leg, you're squeezing blood up when you relax, the blood flows backwards, but the valves close and keep it cooled upward. So it's really important to move your body, get your veins squeezing the blood back to your heart. Here we have another image of our arteries leading to our arterioles, to our capillary bed, to our venules, to our veins. I like this image because it shows you that there are little tiny smooth muscle cells around your capillary beds called precapillary sphincters. That controls where you're taking your blood. So those will tighten or release based on the needs of the cells. If your cells are doing cellular respiration and they need oxygen, you will allow blood to flow through, your little sphincter will relax. If those cells are not really being used, like I'm using a lot of the cells in my top half, but I'm not really using my legs right now, I would have more of my sphincters open in the parts of my body that are doing cellular respiration because they need ATP to be doing all this movement. The other parts of my body might be tightening their sphincters and I'm forcing more blood into the places where I'm using it. So really cool little system. And lastly, I have a suggestion that's a hesitant suggestion. There's an exhibit. It's called Body Worlds. And in this exhibit, they use a specialized technique to preserve cadavers. It's kind of wild, but I wanted to show you these two images. These are a hand and a foot where they used a special type of resin and they inserted it into the blood vessels, all the way through the blood vessels, and then removed the rest of the tissue so that you're just seeing the blood vessels. And it's wild to me because I know that capillaries are everywhere because every cell needs oxygen. They say that capillaries are no further from 3 cells from another capillary. So every cell is only 3 cells away from an oxygen source. And I know that each of these capillaries did not get resin in it, but the amount of capillaries that did is pretty incredible. Like you can absolutely see what these structures are without knowing what these structures are. So here you have a really nice artery coming through and you can see it branching into the arterioles and then down to the fingertips where we have a lot of capillaries. I said I was hesitant to bring this up because it's kind of macabre. It's a lot of cadavers. It's a thing. If you're interested in human anatomy, though, it's pretty wild. Check out the website, see if you're interested in it. It's a rotating exhibit. It has come to OMSI before. It's not planning to come this year, but maybe next year. And if you ever visit the Bay Area in California, there's a permanent exhibit in San Jose. So I went to that one, and honestly, it kind of freaked me out. But it was still interesting, and there's a lot of things to learn. So I don't know, email me. Are you into it or is it terrifying? Either way, I hope you enjoyed this lecture. Have a great day, and we'll talk soon. Bye.